Welcome to another exciting biology podcast by Mr. Mechnick. The topic today is the cell membrane. As we all know, the cell membrane is this structure that surrounds the outside of all prokaryotic and eukaryotic cells. The cell membrane is a semi-permeable structure, which simply means it allows certain materials to move in and certain materials to move out. It has the ability to regulate this, as we will see in the rest of the podcast. So the main function of the cell membrane is so that it can help the cell maintain a stable internal balance, which we know as homeostasis. That's its main goal. So how does it do this? Well, in order to understand how it does it, we need to understand a little bit about the chemical composition of the cell membrane. The cell membrane is made up of a phospholipid bilayer with different types of proteins embedded within it. So both the bilayer and the protein allow certain materials to move in and move out of the cell. So here's a cross-sectional view of what a cell membrane looks like. This is magnified much greater than any type of microscope that we can use in our school to view this. So this is a good model of what the cell membrane is composed of. The tiny circles that we see, these are the phospholipids. They are connected to two fatty acid tails. We call these phospholipids. It's a double bilayer, which means there's a layer of lipids on the inside of the cell and a layer of lipids on the outside of the cell. Embedded within the phospholipid bilayer are proteins. There are many different types of proteins that all serve as a specialized function. So one of the things that I would recommend doing is finding a picture that you could use in your notes or sketch this in your notes and label the phospholipid bilayer and label some proteins so you understand what the composition is. Now, just to give you an idea what the phospholipid bilayer is made up of, the head part, which is made up of a phosphate molecule, so phosphorus with 4 oxygen atoms bonded to it make up the phosphate head. This is hydrophilic. Hydrophilic is a term that simply means that it does like water. So these are polar molecules, and polar molecules, just like water, will be attracted towards it. So we term the head of this being hydrophilic, whereas the fatty acid tails repel water. They don't like it because of their non-polarity, and we call those hydrophobic. So some terminology that you should understand. Hydrophilic means liking water. Hydrophobic means that they fear water. And as a result of this, this particular membrane, when put together, will form a bilayer. The bilayer exists around the entire cell surface, and it's often described as a fluid mosaic model. Reason being is the phospholipids tend to be in motion. They're very fluid. These little pieces are very, very much in motion all of the time. The phosphates or the proteins that are embedded within the phospholipids are constantly in motion as well. Because a cell, the extracellular fluid is simply water. The inside of the cell is mainly composed of water, and those are constantly fluctuating, changing concentrations, and thus allows the movement of materials in and out of the cell. And that's why we call it a mosaic model, because there's all these tiny pieces that would give it the appearance that it's always in motion. So what is so important about this membrane? And I would say there's 5 major functions that you guys should understand. One is that it's very selective. It can select what goes in and out. 2, it has the ability to recognize. So because of the proteins on its cell surface and also some of the carbohydrates, these are identification markers for your body to recognize cells that are from yourself. It also gives your body a way to recognize foreign cells. So it can use the cell surface to recognize normal cells that are supposed to be in your body and foreign cells that your body might want to try to destroy. The other job of the cell membrane is to get rid of waste products. So as your cell is metabolizing sugars, as it's digesting worn out organelles, it has to get rid of that waste material, so it has the ability to do that as well. Obviously, our cells need simple sugars, we need complex sugars, we need amino acids. It has to be able to bring all of those important nutrients into our cell. So there are certain ways that this can happen depending on the type of nutrient. And last, it can change according to its environment. So many different factors can influence how this cell membrane adapts to its environment. So the most common way, or the simplest way that materials can move across a membrane is by simple diffusion, moving with a concentration gradient. So when I use the term concentration gradient, I'm referring to the movement of materials from a high concentration to an area of lower concentration. That's the simplest form of transporting of materials across a cell membrane. So going from high to low concentration. So anytime I refer to a concentration gradient, we're referring to an area where there's a lot of something to where there's an area where there's less of something. And materials will always flow from high to low concentration. Osmosis is a good example of diffusion. However, it's the diffusion of water. We know water is a vital player in living systems, so water can easily diffuse across a cell membrane. The cell cannot stop the flow of water. It automatically diffuses from areas of high concentration to areas of low concentration. So as we learned earlier this year when we talked about concentrations of solutions, water is considered a solvent. Many substances can dissolve in it. Those are the solutes. When you have an equal concentration of water and solute, we call this an isotonic solution. Isotonic solutions means that there's the same concentration of water on the inside of the cell as the outside of the cell. A hypotonic solution is where you have a lower concentration of a solute on the outside of the cell compared to the inside of the cell. As a result, water will move into the cell and the cell will get bigger. So hypotonic, the cells get larger because water wants to move from area of high concentration to an area of lower concentration. And the term hypertonic is referring to a solution that has a high concentration of solute. So imagine salt water as a good example. If you were to place yourself in salt water for an extended period of time, you have all felt it before in an ocean where your skin starts to dry up. That's because your cells were in a hypertonic solution and the water diffused out of your cells, causing your cells to shrink and feel dehydrated. So here's a model that shows isotonic, equal concentrations. Hypo means you have low concentrations in the cell, water moves in. Hyper concentration, high solute on the outside, low solute on the inside. That means the water wants to move from the inside to the outside, and that causes cell shrinkage. So diffusion of water does not require energy. Other molecules like glucose can easily diffuse across the membrane by what's called facilitated diffusion. Facilitated means that we simply need a helper to get molecules across. Water molecules can easily transport through the phospholipid bilayer. Molecules that are too large, that can't fit through the phospholipids, have to use a protein. So these proteins can move materials through the cell membrane from high concentration to low concentration. Again, no energy is required for this. However, sometimes energy is necessary to move materials against the concentration gradient. So that means instead of going from low to high, materials will move from high to low concentration. So in active transport, materials are going to move from where there's less of something, in this case on the outside of the cell membrane, to where there's more of something. So this is going against the gradient, against the flow. And in order for this to happen, energy has to be used to open these transport proteins or carrier proteins to move ions or materials against the concentration gradient. So there are 4 types of active transport that I would like you to be aware of. One is called ion pumping. Ion pumps are referring to moving ions, charged particles, against the gradient. A prime example of this is with your nervous system. We will spend more time in class taking a look at how our bodies can do this and how neurons can send signals so quickly with the use of energy by the use of ion pumps. Co-transport is another type of active transport that requires energy, and a good example of this is when we break down materials in our cells and digest them, and we want to transport them into a cell so we can either get the amino acids, so we can use the lipids, so we can use the sugars. Sometimes these materials have to be co-transported from low to high concentration. And then finally, endo and exocytosis are kind of bulk transport mechanisms. Bulk meaning big items moving across the cell membrane by actually engulfing things. A really good example of this is with white blood cells called phagocytes, how they can actually surround a cell and engulf it. So if you've ever heard of Pac-Man, a game that was popular when I was a young kid, Pac-Man is a good example of a phagocyte that can go engulf food, ingest it, break it down, get whatever it needs from it, and then whatever waste materials are left over, it can excrete it. So exocytosis is the process of excreting cell waste across the cell membrane. It does not require the use of protein channels. It does not move through the phospholipids. It's just the rearrangement of the membrane to allow for bulk items to move across the membrane. We are going to end right there with the podcast on the cell membrane. So again, just to recap, movement of materials across the cell membrane is called diffusion. Simple diffusion is the easiest way materials can be transported across the membrane. The movement of materials from high to low concentration with the gradient is normally how things will move across. The movement of water is called osmosis. Osmosis is the movement of water from high to low concentration. And when we need materials to move from low concentration to high concentration, energy has to be used. So ATP is our main source of energy that will trigger these proteins to open up and to transport materials against a concentration gradient. This was a lot of information in a short period of time. We will cover this a lot more in depth during class period, so please bring questions that you have regarding cell transport to class this week. Thank you for listening.